In 1928, zoologist Ernest Mayr went to Dutch New Guinea's Arfak Mountains. There he recognized 137 species of birds, and also found that the natives had named 136 kinds. When he observed the locals' naming of birds, he saw that both the locals and the scientists were able to distinguish the same species. The ones the locals identified matched the same species as the scientists identified. This leads us to believe that discontinuities in nature are an objective fact. Nature is discontinuous due to the fact that evolution has caused numbers of different species to develop from one single species. These species are discrete groups or clusters that each individual can be classified into. Each group is discrete from each other and have their own organism space. They're discrete as they do not blur into one another. There is variation within a single cluster, but they remain discrete. Bears, for example, fall into different species, each discrete from the other. Note that these clusters are all organisms that reproduce sexually. Another example shown here are species of wild cats: lions, cheetahs, and tigers. Consider the lions. There's variation within the species, but the three species don't blur into one another. They are discrete. The problem of speciation was the idea of how these groups arise. Since evolution is a continuous process, how is it that it produces discontinuous and discrete groups? This was Darwin's problem. He couldn't figure out how the groups arise. He didn't see the discontinuities. He failed to explain the clusters, and he never explained how one single species was able to split and become two. What he did explain was adaptation and selection. This is important when the species splits. There are many species in nature, each with its own set of traits, and they have all derived from one single ancestor. The splitting of one species causes biodiversity, but we need to know how the species split to create a new species. According to the morphological species concept, a species is a group of individuals who resemble each other. The problem with this, though, is that some different species resemble each other really well. A species, according to the biological species concept, is a group of interbreeding populations that are reproductively isolated from other groups. This is the better definition. The problem with the morphological species concept is that species is defined only by how individuals look or by their appearance. The problem is that some individuals within a species don't actually look the same. For example, humans with blue eyes and humans with brown eyes. This concept wants to say that there are two different species, when in fact they are the same. Here are two groups of people: Canadian Inuit and South African tribespeople. They look different, but they can still produce fertile offspring. Another problem is that there are individuals who look alike, but they're not in the same species. For example, there's 9 different species in fruit flies, but you can't tell any of them apart by just looking at them. There's also mosquitoes. They have 7 different species, but they all look the same. How could this be possible? The answer is that, even though they look the same, they can't ever exchange genes. This is because they belong to different gene pools. Species are distinct not because they look different, but because there's barriers that prevent interbreeding. The biological species concept was the better definition because it could explain the fact that species were their own group, regardless of how they appear. It explained how they can reproduce within their species and how they can't reproduce with other species. It's due to their gene pools and how they reproductively isolated. Reproductively isolated means that members of different species are prevented from successfully interbreeding with each other, but members of the same species can. Reproductively isolated also keeps different species from mating with each other. As Darwin believed, species don't arise from the purpose of filling up empty niches. The study of speciation tells us that species are evolutionary accidents. Biological speciation is very similar to speciation of two closely related languages from a common ancestor. Languages can change very quickly when there are very few mixing of individuals from different populations. Human languages change by linguistic selection, when new words or catchy phrases get inverted, as well as through linguistic drift, which is when there are changes because of withdrawal transmission. If you compare language from a few generations ago to today's generation, you'll notice the change by linguistic selection and linguistic drift. The evolution of languages can also be traced back to the past and also a family tree if you notice similarities of words and grammar. New languages, like new species, form as a byproduct of other processes, like the transformation of Latin to Italian in Italy. Darwin's analogy between speciation and languages show that species are evolutionary accidents. Geographic isolation is the first step in the origin of species. It is also known as a theory of geographic speciation. It is important to have the genetic isolation between populations to be geographically isolated. It's important to have them isolated because without isolation, selection that could drive populations apart has to work against the interbreeding that brings individuals together and mixes up their genes. For example, imagine an insect living in the woods that can feed on two types of plants. Each plant requires a different set of adaptations. As each group of insect within the plant begins adapting to one plant, it also starts mating with the insects adapting to the other plant. This mixing keeps the gene pool from splitting into two species and will thus create a generalist species that uses both plants. As shown in the picture below, speciation is much like separating oil and vinegar. They don't want to be together, but will because constant mixing. How does speciation occur? We want to see geographically separated populations turning into new species. In order to see that, we use indirect methods that are testing predictions taken from the theory of geographic speciation. There are 3 predictions that are true and important. The first prediction is that if speciation relies on geographical isolation, populations must have experienced the isolation throughout history. Geographic isolation could have occurred through mountain ranges rising, glaciers spreading, deserts forming, continents drifting, and drought dividing a continuous forest into patches separated by grassland. This can allow species to divide into 2 or more populations. However, this is not the only way species become separated. This can allow species to divide into 2 or more populations. However, this is not the only way species become separated. There's accidental long-distance dispersal. This is when a colony or population can evolve in isolation from its ancestors. For example, a single pregnant female wanders off and ends up colonizing a distant area. The colony will evolve in isolation from its ancestors. This type of isolation is even greater in the archipelagos because individuals can move back and forth between close-by islands, allowing them to become geographically isolated. An example of geographic isolation is Isthmus of Panama. This land formed 3 million years ago, which separated populations of marine organisms. The second prediction of the geographic speciation model states that geographic speciation is still occurring in nature. There are many examples where isolated populations of a single species begins to speciate and show small amounts of reproductive isolation from other populations. For example, in South Africa, there's an orchid that is pollinated by hawk moths and long-tongued flies. To attract them, the orchid develops a long nectar tube in its flower, and only when they get close enough, they can pollinate. In the coastal region, the orchid has short tubed flowers because there's only short-tongued bees. If all 3 types of pollinators were together in an area, the flowers would show some genetic isolation, because short can't easily pollinate long-tubed flowers and vice versa. This is an example where individuals from different populations mate less ready than individuals from the same population. The third prediction is if populations must be physically isolated, we should be able to find the most recently formed species in different nearby areas. You can figure out how long ago species arose by looking at the differences between their DNA sequences, which is proportional to the time elapsed since they split from a common ancestor. An example of this are polar bears and brown bears. Polar bears and brown bears are both sister species and were divided by a geographic barrier. They were diverged about 479,000 to 343,000 million years ago. Scientists and biologists compared the length of the DNA segments shared by polar bears and brown bears to determine when the split occurred. The fourth prediction is reproductive isolation between a pair of physically isolated populations increases slowly with time. Reproductive isolation is when different species may live in the same area but aren't able to interbreed with one another because of isolating mechanisms. For example, some snails can't reproduce because their reproductive parts don't fit. One snail might have a shell moving counterclockwise and the other vice versa, preventing them from creating offspring. Reproductive isolation reduces gene flow.
The primary element needed for speciation is time. Examining diversity of a species reveals that speciation occurs on a spectrum, with some species showing no reproductive isolation, meaning they are a singular species in contact and able to mate. Some species, on the other hand, show complete divergence from an ancestor or sister species with whom they show no reproductive viability. However, most species are somewhere in between. They show some unique traits, but reproductive success is a gray area. The most easily observable cause of speciation is geographic isolation, in which a barrier somehow splits a population and they no longer remain in contact. They begin to diverge according to their respective environments. One example of this type of speciation is the Arctic fox, native to the Arctic tundra, and the red fox in North America. The Arctic fox diverged from their common ancestor during the last ice age, traveling through the then frozen Atlantic. Then, as temperatures warmed, the North American red fox and the Arctic fox experienced geographic isolation and are now considered two distinct species. The idea of one species diverging into two is best conceptualized as a spatial barrier, but it is not necessarily how every species evolved. While developing his theory of evolution, Darwin contemplated a different type of speciation event in which a species could diverge while remaining in contact with its initial population. An obvious complication of this type of sympatric speciation is the presumably continual interbreeding events that would essentially homogenize the gene pool, resulting in only one species. Relative to geographic speciation, sympatric speciation becomes difficult to prove. While it is mathematically possible, observation of biodiversity shows very little traces of sympatric speciation. This is because it is almost impossible to prove that two sister species living in the same area both originated from the area, or if contact was reestablished after a localization event. Evidence for sympatric speciation can be found in certain areas which are extremely small and are isolated from other sources of gene flow. Habitat islands become the ideal model to recognize cases of sympatric speciation. The best example of sympatric speciation comes from lakes in Cameroon. Each lake lies in its own isolated volcanic crater and ranges in size from 1 to 2 miles. The lakes contain many different species of the cichlid fish, all derived recently from their common ancestor, and each in its own distinct species group. Even though on many occasions sympatric speciation could have occurred, it appears to be a rare event. Numerous islands have been observed and examined for sympatric speciation, but the vast majority of them do not show any evidence for sympatric speciation. This is due to the initial dilemma of continued interbreeding and homogenizing. This is a reasonable prediction of speciation. Speciation usually requires many generational times to view evidence of divergence, but there is a type of speciation that can be witnessed in a relatively small span of time. This speciation occurs in autoploidy organisms, the most common of which are plant species. Two different species of plants can mate, and although they differ in chromosome number, a duplication event solves this discrepancy by doubling the chromosome number. This allows for each chromosome to pair with itself and produce viable offspring. Polyploidy speciation is not a particularly rare event. This is because plants produce millions of sperm and eggs, significantly increasing the chances of autoploidy mutations. The best evidence of this type of hybridization producing new species is the recent speciation of the groundsel Senecio cambrensis. The groundsel Senecio vulgaris, with a 20 count chromosome, is native to the UK. The ragwort Senecio qualidus was introduced in 1792. Around 1910, a new species was documented to have appeared and subsequently proliferated. The new species was proven to be a hybrid of the two through crossbreeding in greenhouse experiments.